Welcome to episode 4 of Health. Welcome to episode 4 of Metabolism, Diet, and Health. I'm Hunt Bobo, MD, and this time we're going to talk about enzymes. What's an enzyme? An enzyme is a nanomolecular machine that does all the metabolic processing, and there are multiple good references. This is the most common one I've found in the literature now, is a Japanese reference that shows all the different metabolic pathways. My favorite enzyme and metabolic pathway to be done is the Roche biochemical pathway. Biochemicalpathways.com. I'll put a link down below in the additional material. But this is my favorite one. It's like a roadmap of the world or United States. It's got 2 big parts. It's not complete, but it does include bacteria, plants, and photosynthesis. This is the pathways. This is the cellular structures that contain those pathways. And as before, we can always look up G6PD, glucose 6-phosphate dehydrogenase, and may have to spell it out to get it to work. Nope, so I'll spell it out. Glucose 6-phosphate. Phosphate. Okay. Glucose 6-phosphate dehydrogenase. And we just tap the number, and it'll take us to that. We can remove some of these, get rid of that side label. We can go to number 1, where that was, right here. Hate to do that, clear the search so we can see. But glucose 6-phosphate dehydrogenase is a fairly common genetic defect that we have. And this is we'll go back to the other one and show the pictures of the enzymes. The enzyme means in yeast. The Germans took it from the ancient Greeks. Zyme was a German word for yeast, which came from Greek word zyme, and it was first found in yeast, and it's a protein made of multiple polypeptides, which are made of multiple amino acids each, all folded together in a tertiary structure to act like a nanomolecular machine that lowers the energy of activation of converting one chemical to another, converting the glucose to glucose 6-phosphate. You have to add in a higher energy molecule like the NADP plus up there. Enzyme. Okay. Delete the rest. EYME. Okay. Wikipedia. Enzyme. I looked up the references for enzymes, and some little educational sources said there's 1,300 active useful enzymes in the human body, and other sources said there's over 75,000 enzymes identified thus far in a human body. Enzymes come and go. We have fetal hemoglobin before we're born, has a higher affinity for oxygen, so it can suck the oxygen out of our mother's blood from the placenta. And when we're born, the bone marrow immediately starts taking over the production of adult hemoglobin, which has more appropriate oxygen binding capacities for breathing air. And then the heart does some remarkable things, closing up right to left shunt through the atrium and right after birth. And then patent ductus arteriosus is closed spontaneously, or sometimes with a little help from maybe an NSAID. But the enzymes constantly evolve and come and go. In Africans and in equatorial people, there's a high percentage of lactose intolerance past childhood. And the further north you go, it seems like the less they're affected by this lactose intolerance. But supposedly, there are, Scandinavians even that have lactose intolerance that hits them on up in their late middle age or early old age. And that's because the lactase enzyme that the babies have, all babies, all African, South American, and Scandinavian and Chinese people have, just quits being produced when you're around 5 to 10 years old. Probably more around 5 for the true lactose intolerant people. The same thing happens with our ability to manage sugar. Adult onset diabetes, they get too fat and get on up in their 50s and 60s and get to where they become diabetic, and they were never diabetic before. But enzymes are constantly turning on and turning off throughout life. I mean, there's a whole complex set of reactions that go on with menopause, from ovulating every month and having cycles to becoming postmenopausal. And the hormone productions in the tissues in the body don't produce the same type of hormones, and a whole different metabolism takes place. So expect the enzymes to come and go and change. And for one given enzyme, there may be multiple different forms of the enzyme. And the same thing can be said for a similar proteinaceous structure, the membrane receptors, which are basically binding sites that can transport in or out, or else they're a combination of a receptor and an enzyme. For instance, the cyclic AMPase, which takes ATP, the universal energy molecule, and makes it into a cyclic compound, which is the second messenger that you use when you have epinephrine or norepinephrine. The cyclic AMP is broken down by cyclic AMPase, and this cyclic AMPase is inhibited by the methylxanthines, caffeine and theobromine and theophylline. And we get jazzed up in fast heart rate and jittery when we drink too much coffee. And that really is an enzymatic inhibition. We're inhibiting the enzyme that breaks down the second messenger cyclic AMP. But this is a picture, a cartoon drawing of an enzyme that tabs. I'll have to pull it back up, and we'll go to my talk here that I put together when I first started trying to do this. First, an amino acid is the basic building block for proteins and for many of the other smaller structures. But this is an amino acid with a carboxylic acid group on the right here with a double bond oxygen and a single bond oxygen hydrogen, which is COOH, is a carboxylic acid group. The alpha position is the first carbon after this carboxylic acid carbon. There's an R that's attached to it that basically determines what type of amino acid you have. We basically have 20 amino acids in our body that are alpha amino acids. The nitrogen up to the left in here is the amine group to make it an amino acid. Amino acid doesn't necessarily mean it's an alpha amino acid, but this is what we're talking about when we talk about amino acids in human beings. There are actually like 22 or so that they found in humans, but one of them is not really used by humans, and the other one is not really necessary. I'm going to go over those in just a second. So that's an amino acid, and there is a linkage between the alpha carbon amine group and the carboxylic acid on the next one. So there are 9 essential amino acids that we absolutely have to take in from the environment, period. Histidine, leucine, isoleucine, lysine, methionine, phenylalanine, threonine, valine, tryptophan. The side groups all have different characteristics from just being a hydrogen in the case of glycine, or a methyl group in the case of alanine, to complex ring structures like tryptophan or phenylalanine. 6 other amino acids are actually essential in small, rapidly growing children or sick people, and they probably help maintain a vigorous life that you wouldn't have otherwise if you didn't get any of these, and make activity and healing and resistance to infection easier. This is arginine and cysteine, glutamine, proline, tyrosine, and glycine. Now, glycine is also a neurotransmitter in the correct locations, and it's the shortest and smallest of all of them. Seems like we'd have an enzyme that could just cleave off the methyl group off of the alanine and make glycine. 5 amino acids seem to be so easily made from other substances that they're not considered even conditionally essential. And this is alanine, which is like a glycine with just a methyl group on the side, R group. Aspartate, glutamate, serine, and selenocysteine. This is a peptide bond. So we have the carboxylic acid group on the right of the middle agent, COOH. Then we have this red nitrogen on the left side of that second group, which is actually, it's R, doesn't tell you what it is. The R group is just representative for some type of organic compound that determines what the name of that amino acid is and what its physical characteristics are. On the left-hand side, the very first chemical in this structure, it still has the R sticking up, but it's been flipped down so you can see how they link together. You take a water out of this group, infuse the amine group on one side to the carbon of the carboxylic acid group on the other side, and that's a peptide bond. And this is what happens over here on the far right. The water comes off, and you have a peptide bond where you have a double bond oxygen, nitrogen, then a alpha carbon, then you have the carboxylic acid group. And this carboxylic acid group will just hook up to another amino acid to where you string along 10 or 20 up to maybe 60, and you call them polypeptides. An oligopeptide, I think they usually talk about something maybe less than 10 or 12, but they're polypeptides, all of them. And you get 100 or 200, and you have a big long peptide chain with certain amino acids in it, and it folds up in a certain way, and you get 4 of these, and you have hemoglobin. Then you have this glucose 6-phosphate dehydrogenase we've been talking about. Peptides do a lot of different things. The smaller, shorter ones are hormones. Insulin is a peptide. Oxytocin, the mother pair bonding or mate monogamy hormone and the lactation hormone, letdown hormone, and the salt water balance hormone, vasopressin. Those are all just polypeptides. In endogenous antibiotics is one class of polypeptides in bacteria and fungus. And everybody's heard of endorphins and enkephalins. Endorphins are peptides. And then stuff that makes your intestines move right, the vasoactive intestinal polypeptides and things they check for congestive heart failure, brain natriuretic peptide, and for head injuries and controls your salt and water balance. There are many, many more polypeptides. And in, with doing pituitary surgery, as I'd said before, we have these enzymes cutting on and off that are produced at one stage in life and then shut off, sometimes to our benefit, sometimes to our detriment. There are polypeptides that are being produced that are just junk, that the cell has lost its ability to produce the proper peptide. We take out pituitary hormones or pituitary tumors that were growing. The great big ones weren't producing anything that actually did any hormonally active function, but they weren't just sitting there not producing anything. They frequently produced screwed up growth hormone or screwed up thyroglobulin, screwed up polypeptides that just weren't quite right, weren't quite the same. They were mutated and still being produced, but it wasn't quite right. And this is a common theme over and over again. If something becomes useless and you have to replace its function and it's still there, it can actually still be produced and develops more and more errors because the errors are not self-correcting. The organism can go on and survive, thrive, and reproduce, and it gets passed down till you accumulate more and more errors in your DNA for that one particular protein. This is a picture of pork insulin. We were saying that was a polypeptide. See how it's folded up. I make no apologies for cartoons. We actually can't do a good job of visualizing these things at this magnification. It's in a solution and is constantly vibrating and Brownian motions moving all over the place and diffusing, and it's folding and unfolding with shocks and heat and being passed and moved around, or when it engages into its receptor. So these are just things to help you get a concept of what's going on, but not really a good clear photograph or picture. So cartoons are okay to help understand what's going on, to try to draw it like blueprints. Proteins, they're longer and bigger, something longer than 50 amino acids. And they're frequently bonded across cysteine bonds and other type of interconnections that help maintain the three-dimensional structure. Examples of proteins are enzymes, collagen. Elastin is the stretchy yellow ligament that's in your body. And, receptors, membrane pumps, all those things are collagen. Your dermis is mostly collagen. Not the receptors being collagen, but they're proteins. And this is a three-dimensional picture in 2D of hemoglobin, which has iron in it. It's got 2 alpha and 2 beta strands, so it's got 4 long polypeptides with this heme ring right down there, the little flat thing, kind of a flat planar ring like cholesterol. So these are amino acids in the ribbons, but down inside of each one of these alpha rings is a heme that has an iron right there in the middle of it, the little jack-like thing surrounded by blue stuff. So that's a representation of a hemoglobin. And, well, try to get rid of that thing. Here we go. This is a representation of a collagen with 3 different polypeptide strands. There are multiple different types of collagen, and then there are defects in this since we're all different. One name to come out would be Ehlers-Danlos syndrome, which basically is the rubber man at the circus that can fold up and get into a basketball or fold out or stretch their neck skin up over their head. They have elastin, but the collagen's not very strong, and they end up getting aneurysms or their lenses get dislocated. Then there's some people that just have very tough inelastic collagen and break. There's all kind of disorders associated with having the wrong material for your connective tissues, and it's not just your Achilles heel or your anterior cruciate. It's your abdominal aorta and your lens in your eye and lumbar discs, and your cardiac skeleton with the mitral valve and aortic valve. And that's a vague and poorly studied subject that people are working on. This is a good representation of the nano machine that is in the mitochondria. It makes adenosine triphosphate. You know, there's a portion that's inside the membrane, which is basically a lipid bilayer, meaning it's fat soluble. It's a layer of oil. And then the aqueous cytosol inside, which is polar and would be something that would be more water soluble, like water solution. And this is what a transmembrane anchored membrane-based enzyme would look like, and it helps to make your little factory to process NADH and electron transport down to make ATP as it's going. This is an example of a fatty acid that's my favorite fatty acid. It's really not essential according to most sources, but it's a conditionally essential amino acid. And this is docosahexaenoic acid. It's beautifully symmetric. On the left, over here, the carboxylic acid group. C is where the 1 is. OH is the carboxylic group. And that's 1. Then the next carbon is at the next angle. 2, 3, 4 is written in blue. There's the first double bond from the carboxylic acid in. But when you're talking about omega 3 and omega 6, we're not talking about from 1. We're talking about from the end, omega. That will be the alpha back in here, and on the carboxylic acid end, the omega is the end. So this is an omega 3. The first double bond starts at the 3rd carbon from the end. You'll see it labeled over here in red. It's omega, is that W looking letter. There's 1, 2, 3. The key thing about this is these are anti-inflammatory fatty acids. They're not metabolized into the prostaglandins that cause your joints to swell and ache and cause things to clot up like some of your leukotrienes might. And your omega-6 fatty acids, which we'll go to next, actually are the precursors for this inflammatory response. Now remember, you don't want to absolutely get rid of all of your inflammation. That's like having HIV and dying from an immunodeficiency. You need to have the ability to fight infections and have inflammation. If you don't, you die young. And it's a balance. We want a balance to be able to fight infections when we need it, but not to be hurting and eating our joints up. So docosahexaenoic acid, DHA, comes from Arctic krill and cold water fish. The more double bonds in a fatty acid, the lower the melting point, the longer it stays liquid. If you have a shorter fatty acid, it melts quicker. An example of this process would be methane boils quicker than ethane, quicker than propane, quicker than butane, and hexane, 6 carbons. Octane, which is about like gasoline, even though gasoline's actually a mixture, but gasoline's a liquid at room temperature. So the longer the chain of carbons, the higher the melting point, the higher the boiling point is, and the more double bonds. If you have palmitic acid or 20 carbons and you put 2, 3, or 4 double bonds on that 20 carbon chain, the more double bonds you put, the more curled it gets, and effectively shorter it is, in the longer it stays liquid as you cool it down. The quicker it melts when you have it in a solid form and you heat it up. The quicker it boils. The melting point decreases with increasing number of double bonds. And that's why the best source of this docosahexaenoic acid is your Arctic cold ice water fish and stuff like that. All these enzymes are coded in their polypeptide sequence by DNA, and this is what the basic DNA double strand pairing looks like. Thymine versus adenine. The whole structure is thymidine and adenosine, and with the phosphate it's adenosine monophosphate, or thymidine. Here's got the sugar backbone. That's a ribose group right next to the little 5-sided figure, and then the phosphorus links them together. Guanine pairs with cytosine. They have a little stronger bond because they've got one more bond than thymine and adenine has. All these organelles are biochemical pathways are either free in the cytosol or organized by these little organelles: mitochondria, endoplasmic reticulum, nucleus. And this is the target of statins like simvastatin and atorvastatin, lovastatin. It's HMG-CoA reductase. It's kind of a big complex molecule with a bunch of different colored ribbons on it. And the statins keep it from working. And it is listed right here on the Roche biochemical pathways. It's hydroxymethylglutaril-CoA reductase, and it has a participation in taking, the 3-hydroxymethylglutaril-CoA to mevalonic acid, and back and forth. And there's palmitic acid, vitamin A, and retinol and retinoic acid. All this is in your steroid synthesis in your liver. And there are people that have enzymes that work in such a way they produce too much, don't have the control, produce too much steroids, and need to have a statin. But then I think a lot of the people that are getting them now have muscle pains and muscle wasting from the statins, and may be better served by going on a fast one day a week and getting more exercise and cutting out all the carbs. This is my little scribbling and pretending to draw a lipid bilayer. The red is the carboxylic acid end of these triglycerides and phospholipids. And the blue is the long fatty acid that's oil, that's hydrophobic but lipophilic. The blue thing is my drawing of a transmembrane protein. You see where I put in the COOH? COOH is carbon double bond oxygen, single bond O, single bond H. And then the blue is just a bunch of carbons strung together for however long. And then I drew a cell with mitochondria. And this is the pathway that I will just flip over to the here and look at. But I love this thing to be able to look up and see what's going on. There are a bunch of them out there, but this one is a good start for me because it's quicker to load up than the other ones, the KEGG pathways. So thank you for joining me. I hope you've learned something about what an enzyme is and what amino acid is and a fatty acid. We'll come back and go into this in detail later, but remember, try and eat as natural food as possible, as little processed as possible, but use common sense. I wouldn't get chicken or ground beef from the grocery store and eat it raw. I think you're asking for food poisoning. And I wouldn't go out and shoot a wild pig and eat it raw. I'd cook those well and cook your meat well enough to where it's tender enough to eat. You will lose some of the vitamins, but you need to eat some fresh fruits and fresh greens if you're in your own environment that you can take and keep the vegetables clean. I would not go down to Mexico in an area that has possibility of amoeba or shigella. A friend of mine, his mother got shigellosis eating a salad down in Mexico. I've been to Mexico many times and never gotten sick, but tended to eat cooked stuff. I did have some ceviche, and it was probably the best food I'd ever had in my life, which is raw fish with lemon and lime on it. But then, I could have gotten something like I got in the Grand Canyon when I got giardia, even though I was not drinking the water. I don't know how I got it, just, I guess, swimming in it or something like that. Cook as little as possible, eat as fresh as possible, and use common sense, and keep your diet as wide-based and varied as possible. Don't just stick to one thing like hot dogs and American cheese or just leafy greens. You got to vary it. You need the folate from the leafy greens and, maybe a little red meat every now and then to get the proteins you don't get as much otherwise, and plenty of fish and plenty of legumes and whatever you tolerate. But some people, they can't eat beans, or some people can't eat this or that. You have to use your family history and your local knowledge. I'll see you next time. Bye.